So we're here to talk about oxidizing disinfectants, and they're used in many, many applications all over the world, but particularly we're going to be looking at food and beverage. Those terms, food and beverage, are really incredibly generic umbrella terms for a wide variety of sub-applications, whether it's meat, poultry, and seafood, or ready-to-eat bagged salads, through to bottled water and soft drinks, even brewed products like beer and kombucha. The term food and beverage encompasses a very wide variety of products and commodities. And of course, when we think about how oxidizing disinfectants are used, not only do we have those wide variety of products and commodities, we have a wide variety of application points where and how we apply those disinfectants throughout the process. So we've got irrigation disinfection all the way at the beginning on the farm, through to tools and surfaces. We've got chillers and dips, sprays and flumes, it goes on. And a lot of those application points will be present in many different ones. So for example, sprays is present in both beverage and food, although irrigation, you might argue, is just food. So wide variety, each one with their own requirements and conditions. And then just to add further to our options, we have a wide variety of disinfectants that we can apply. So we've got traditional ones like chlorine through to more modern ones like peracetic acid, and ones that I haven't even listed like hydrogen peroxide. So we've got a lot of options, a lot of spinning plates to keep an eye on. We've got the commodity in the product, we've got the application point, and we've got the chemical that's being used that we all need to pay attention to when we're thinking about advancing our disinfection processes. And we often go into plants and see that we have multiple different disinfectants applied at multiple different points. So we might have a dip tank that uses chlorine, and then that same commodity downstream will then get sprayed, but with peracetic acid. So there's definitely a lot to keep track of. And it might seem an obvious question, but why do we use disinfectants? And the obvious answer is product safety. So disinfectants are there to kill microbes, making sure that our products are safe for our consumers who consume them. But there are other benefits to that as well. The use of disinfectants to reduce microbes means that we also see shelf life improvements and sensory improvements in our products. It can increase our product traceability. And of course, if we do all that right, if we've got safe, shelf-stable products in the field, that protects our brand reputation. And there are also financial implications as well. Disinfectants cost money, they cost water. All of our processes have a financial cost associated with them. So it isn't just a case of why do we use disinfectants, but why do we spend time and effort making sure we've got that process as optimized as possible? Well, there's a significant implication if we get it wrong. One thing I'd like to take a second to talk about, maybe not in the kind of natural flow of a presentation like this, but something that always catches my eye, is the public perception of the use of disinfectants in food and beverage processing. And this is the slide where I do have to say that everything I say on this slide in particular, these are my own opinions. They're not the opinions of Palintest or Halma, but I thought I'd share them nonetheless. There is a lot of chemophobia, a lot of fear of chemicals when it comes to food and beverage and the products that we eat and drink. And of course, this is often at odds then with the desire to constantly improve quality, to constantly reduce recalls. So it's an interesting crossroads that everybody in the industry has to kind of manage. And I'd like to really point out one example that, as a Briton, now living in America, I've kind of not really been able to avoid for the last 2 years, is the UK media in particular latching on to low standard American chlorinated chickens. And the British media has been pretty interesting in this. They've, media from all sides of the political spectrum have latched onto this story as when the UK and the US agree a trade deal that we'll all be forced to take these chlorinated chickens. And they've really strongly implied that the chlorination itself is inherently bad, that the chlorine is some kind of nasty chemical that we don't want associated with our food. And of course, this ignores many facts. For one, the vast majority of chickens in the US aren't chlorinated. Most of them are treated with PAA. And the fact that every bagged salad in the UK is rinsed with chlorine. Every ready-to-eat produce, or almost every ready-to-eat produce, is chlorine rinsed in the UK. So, we're already chlorinating food. The reason that Europe doesn't chlorinate meat or apply any kind of direct intervention oxidant to meat is the EU believe that allowing direct intervention disinfectants during processing then fosters poorer conditions on the live sites, whether that's the hatcheries or the farms. They kind of say that if we've got this safety net at the processing side, well, that then would just encourage the farmers and the hatchers to be more relaxed about their standards. And again, my personal opinion, I don't think that argument holds water. You know, to me, it's like saying, let's all drive without seatbelts because that will make us more cautious drivers. You know, I think you can have high quality standards on the live side and you can legislate and enforce those, and you can have additional safety nets in the processing side. And when you look at the number of chickens on UK supermarket shelves that test positive for Campylobacter, it's clear there's a lot of improvement on all sides of this. So I think that's always something that's worth talking about, that public perception is as industry insiders to some degree, understanding how those consumers perceive what we do is important. Back to our regular schedule, and we have to disinfect. We know we do. It's just a case of which one we choose and how do we apply it. And whichever one we choose, we need to measure it. You can't effectively apply a disinfectant if you aren't measuring it. And there are two ways to do this typically. We have offline methods of analysis where you collect a sample and you do a measurement and you take that number and perform an action with it. And we have online measurement where you might have a probe or a sensor installed in situ that's continuously reading, and then the output of that sensor may then control things like your dosing pumps. So of course, we just all use the advanced online automated systems, right? Well, even those systems require regular and oftentimes surprisingly regular calibration, and we have to calibrate them with an online method. So we still need to pay attention to that because what you use to calibrate your probe is critically important. I mean, after all, an online system will only ever be as good as its calibration. And then for food and beverage in particular, a lot of the probes just aren't practical or they don't exist. You know, food and beverage water is interesting. It has a lot of organics. And when you have a fixed probe, it tends to foul incredibly quickly. And that fouling degrades the calibration. And oftentimes, that degradation is so fast, it's not even practical to have an online technology in place. So a lot of food and beverage processors rely entirely on offline methods. So what offline methods should we be using? Well, ideally, we'd all be using the gold standard lab methods like HPLC, high performance liquid chromatography, or full titration. We'd use these because they're highly accurate. They don't have many interferences. They're trusted. You know, we call them gold standards. But they're slow, takes a long time to get a result, they're expensive, they're complicated, and they require highly trained staff. And when we think about food and beverage operating environments, these are manufacturing facilities, really, and those techniques aren't suitable for that environment. So instead, we rely on simplified technologies that speed up the measurement and mean that those less skilled users can accurately perform them. And we've got kind of a few different technologies that we rely upon for field level analysis. We've got amperometry, which we'll talk about in a bit. We've got drop counts, which are kind of miniaturized titration kits. We've got photometric and colorimetric systems. And then we've got something even as simple as a test strip. Now, while I could talk to you all day about this, unfortunately, we don't have all the time in the world. So I'm really going to focus on just one of these scenarios. I'm going to focus on peracetic acid and looking at the drop count kit in comparison to amperometry. And when we think about advancing disinfectant analysis, advancing our processes and our control, it's really important to stop and take a moment to understand, well, what are we doing now? How does that work? What are its limitations? Because if we don't identify those, we can't then identify the correct areas to implement improvement. So I'm going to take a few moments just to explain how a drop count kit works in regard to peracetic acid. So in a drop count kit, you take your sample, usually about 10 milliliters, that contains your peracetic acid, and you add potassium iodide, which is clear. And the idea is the peracetic acid will react with this iodide and turn it into iodine, which has a deep yellow-orange color. The more peracetic acid you have, the more of these reactions occur, the darker your sample goes. In the next stage of the test, we then add starch. And starch is there to change the color from orange to blue. It binds to the iodine and changes that color. And this is important because in food and beverage processing waters, our water's kind of already yellow or quite highly colored to start with. So going from an orange to a yellow color is quite hard to detect in the test, but going from blue to yellow is much easier. So that's why we add starch. Then we add our titrant, which is the last stage. Usually this titrant is thiosulfate, so we add a drop that introduces the thiosulfate into the sample, and the thiosulfate then reacts with the iodine and turns it back into iodide, which is the colorless compound. And we keep adding drops until all of our thiosulfate has reacted with all of our iodine, and our blue sample goes colorless. We count the number of drops we had to add for the sample to go colorless. That means you had to add, the more PAA you have, the more iodine you have, the more thiosulfate you had to use to make it go colorless. So we can correlate those. We can correlate the number of drops to a parts per million concentration of peracetic acid. But there are some fundamental chemical limitations to this. The first is hydrogen peroxide. So anybody using PAA in a food and beverage setting will know all blends of PAA are made of equilibrium mixtures of the peracetic acid, hydrogen peroxide, and acetic acid. And for many mixtures, the hydrogen peroxide is actually present at a higher concentration than the PAA. So this interference can be very significant. And it interferes because hydrogen peroxide will also react with iodide and turn it into iodine. This makes our sample darker. It means we have to add more thiosulfate, which we then interpret as having a higher concentration of PAA. Now, this reaction is slower, but can still be very significant depending on the time of your test and the amount of hydrogen peroxide you have. So there are a few different ways we can try and overcome this. The first is to remove the hydrogen peroxide before we even do our titration. So if we get rid of the hydrogen peroxide before we add our iodide, then we don't have to worry about the interference. And this is exactly how those lab titrations work that we referred to earlier. We use either cerium or permanganate. They react with the hydrogen peroxide and destroy it. And once all the hydrogen peroxide has gone out of the sample, we can then carry on with our test as normal. So why aren't we all doing this in the field? Well, it's complicated. If you add too much of your cerium and permanganate, you can actually make your results worse. There's a temperature control issue, and it often requires special chemicals and glassware. So it's not practical as the real answer. But that's what we mean. When we say titration, that's what we mean. We mean that two-stage titration. And there's often a bit of confusion in nomenclature around these. A lot of people will refer to the drop count kits as titrations. It's the same technique, but it is a cut down version that almost always skips this hydrogen peroxide removal step. We can try and compensate for the reaction. So if we know this reaction is going to happen, we might be able to then anticipate it and then compensate for it in our calibration. So we might say if we have 5 ppm of PAA and we measure 7 because of the hydrogen peroxide, well, we can just build that calculation in. So it'll read 7, but we now just compensate that out and say, well, instead of saying 7 ppm per drop, let's say 5 ppm per drop. And that works quite well in clean water standards. In a clean water standard where you take just PAA and just pure water, you can accurately predict how much that's going to happen. The issue occurs when you go to real water, when you go to real processed water that is not clean, is full of organics. And what occurs is the peracetic acid and the hydrogen peroxide ratio don't stay the same. In some waters, like fresh produce waters, particularly where you have recirculation, you can build up your hydrogen peroxide to very high levels so that the compensation you've applied isn't really enough and you still get high results. In meat processing waters, red meat and poultry, the opposite can happen. You can actually consume your hydrogen peroxide faster. So now your compensation's too big. You're compensating for hydrogen peroxide that isn't there, and now your results are low. So that's an issue. And 3, you can perform the test quickly. Because this reaction is slow and it's continuous, the quicker you perform the test, the less of these reactions occur. But of course, the downside is the more you rush the test, the more likely you are to introduce errors at other points. Another issue we have with these kits that we see in the field is that many users use the low range or the regular range kit even when they're dosing high concentrations of peracetic acid. So you do end up at a point where around about 600 ppm, bit higher for some kits, you just run out of reagents really. So it could be the iodine, it could be the starch, it could be the buffer that you add, but you end up reacting all of the chemical in there and you've still got peracetic acid left over, but now it's got nothing to react with. So you eventually just stop counting it really. And that's a big issue. And we do really see that happening. So here's some data that we have from a customer showing this occurring. So you can see the black line is the expected result. That's what we should be seeing. The blue line is the Kemio technology that we'll talk about in a bit. But what I'd like to highlight is the orange line, which is an industry standard drop count kit. And you can see as we get above 600 to 700 parts per million, that begins to deviate quite a lot. And it might not look like much given the scale of this graph, but if you begin to look at the numbers, you'll see that 1000 ppm of PAA and 1200 ppm of PAA only show up as 800 and around 850 on the drop count kit. So you've moved up, you've got a 20% increase in the amount of chemical you're dosing. That's only showing up as a 5, maybe 6% increase on your test method. When we consider the cost of some of these chemicals, that's very significant, and it makes it very difficult to process, control, and really understand what's going on in your water when you're using a method that isn't really suitable. And these manufacturers know that. They provide high range drop count kits specifically for this reason, but they tend to be such high ppm per drop. They might be 100 ppm per drop, even up to 300 ppm per drop, that they're just not practical for the process control in these applications. So those are kind of the chemical issues. There's also the practical issues with these kind of methods. Anyone who's used a drop count kit will probably be aware of these. For one, you have to hold the bottles in the correct orientation, because if you hold them at the side, that changes the size of each drop and therefore the amount of chemical that you're adding. The one that always gets me is the one in the middle. Every time I do it, I end up doing this. It's very easy to build up static electricity on the plastic container that you do your test in, and you go to add a drop. And if you're not dead on center, the drop will actually be attracted to the static and it sticks to the side of the container. So now you've added a drop, but it hasn't gone into your sample. But then you're not sure, well, if I swirl it, will it then capture it later? It's something that always gets me. And speaking of swirling, something else we see in the field are users who don't swirl their samples effectively. They don't wait for that mixture to finish happening, which means they can keep adding drops, even though if they just waited, the color change would have happened. A couple more. We've got something called endpoint creep. So endpoint creep is where you add a drop of your titrant and it goes colorless, but then a couple of seconds later, that color comes back. And so you add another drop, and a couple of seconds later, the color comes back again. And it feels very difficult to determine, well, where is my endpoint? Was it 10 drops ago when I first saw the color change, or is it now that the color change is actually staying there for more than a couple of seconds? And the reason that this occurs is because of the hydrogen peroxide. We mentioned it's there at a high concentration, and that reaction is slow. And because it's slow, it means that basically it's just continuously churning away in the background, generating new iodine, which is why the solution goes blue. So that's often a challenge, and even an expert user can really struggle with endpoint creep. The other thing we see, particularly in busy plants, are people who've got just a lot of things to do. Testing is just one part of their job. And they tend to do the test something like this, where they had a very, very rapid stream of titrant just to get the test over with as quickly as possible. I think there's a little bit of human nature in this as well. If it was 60 drops yesterday and it was around 60 drops the week before, well, I'll just quickly add 60 drops now, and so long as I see a color change, it will be okay. So we do regularly see that. If you're doing a test as per the instructions, you're adding a drop, you're swirling, you're waiting, you're adding a drop. A high concentration drop count kit should be taking you several minutes if you want to get as an accurate as possible result. And sometimes we see that cut down very significantly, often at the detriment of accuracy. Then we've got the traceability issues, something that is maybe not as obvious straight away, but still a big deal. So a lot of the drop count kits will have larger refill bottles just because it's more economical. We've been on sites where we've literally seen people fill up, put a reagent B refill into a reagent A bottle. That happens surprisingly frequently. We've seen, because of using refills, the small bottles just get reused over and over again, and the labels get wet and they peel off. And if you can see the expiry date, is it actually the expiry date of the refill, or was it the expiry date of the original solution? So that becomes an issue. And then the final one is, I think, the one everyone's aware that can happen, even if it doesn't always, but I like to call it a flat line syndrome. You look at the results and it's been exactly the same result every week in a row for the last 6 months. You do begin to question, how authentic is that? So for us, we wanted to try and advance the chemistry of the method, the fundamental how does the method work? But simultaneously, we wanted to remove those technique errors because one without the other doesn't really advance. So we spent a lot of time on this, and we ended up settling on amperometry. So amperometry is the measurement of electrical current. And in an amperometric test, we generate an electrical current specific to the analyte we're trying to measure, in this case, peracetic acid. So the idea is the PAA reacts with an electrode, generates an electrical signal. The more PAA you have, the more electrical signal, the more current you generate, and we can just read that on a meter. And this is the technology that almost all of the online probes use. It has a lot of benefits. For one, it's resistant to hydrogen peroxide, so we don't have to worry about that hydrogen peroxide interference. It doesn't matter about the sample color or turbidity because we're measuring electrical current and not a color change. We don't care how colored that sample is. But there's an issue in that with online probes, as we've already mentioned, they foul very quick. So although the fundamental method works, the actual practical application in the field isn't always great because the calibration drifts too quickly. So we really wanted to capture the benefits of amperometry, but overcome those negatives. And that's where we come up with disposable amperometric sensors. So a typical amperometric probe might use a precious metal for its electrode, or some kind of fixed metal, could even be zinc, but it could also be platinum. For our technology, we found a way to generate that same selective current, but using carbon. By using relatively cheap carbon, we can make our sensors single use and disposable. So single use means you're changing the measurement surface every single time. Now you don't need to worry about fouling. Now there is no calibration to drift because it's a brand new sensor every single time. So we really have overcome the fundamental limitations of amperometric probes that you see in food and beverage situations. And if you look at the graphic of our sensor on the left there, you'll see each sensor has two electrodes. It's got a low range and a high range for PAA, but we can use those two electrodes for other things on other tests. So for chlorine, for example, one of the electrodes measures free and one of the electrodes measures total at the same time. So within the time scale of the test, you actually get two results, two measurements. But that would not be of any benefit if we didn't also work on the technique dependency. So, we've got that improved chemistry method from the amperometry, but if we can't make it easy to use, we would still introduce errors from operator technique, and we've wasted all that work. So for us, we spent probably as much time on how do we do the test? How does the user interact with the test to try and remove as many sources of error as possible? So we start with scanning the sensor. This makes sure, am I using the right sensor? Has it expired? We're checking all of that for you. So you scan your sensor, you fill your sample vessel, you insert the sensor into the instrument and you close the lid, and then the instrument takes over and does everything else for you. And in 60 seconds, you'll have your result on the screen with no interpretation required. So for us, we really have captured the same benefits as online technology. We've got no hydrogen peroxide interference. We don't have any chemicals or glassware like you do with a drop count kit or other methods. Because we're looking at electrical current and not light, we're not affected by particles, by color, by organics, fats, oils, and greases. The result is objective. So it doesn't require individual interpretation, and it has much fewer steps than traditional techniques. It means less user skill is required, less training is required. Because those sensors are disposable, they're not affected by fouling or calibration drift. We're changing that measurement surface every single time. And because it's digital, and because we're logging the results in a protected file on the device, we have full traceability should an auditor come and want to see our records. But there is an increased cost. Having a single-use advanced technology sensor will cost a bit more than a traditional drop count kit. However, when we consider the cost of the chemicals that we're using, often that increased process control will pay itself back very quickly, and Kemio is able to show and prove cost efficiencies and return on investment in most use cases. So how did we validate it? How did, we had this technology, we wanted to release it to the market, but we have to be able to prove that it works. So what steps have we done to do that? So one of the first things we did was work with a group called CEBAS in Spain, and they are, I think it's University of Murcia, and they are specifically focused on food and beverage processing research. So we asked them to help us compare our PAA sensor, which is the turquoise dot on the left to a gold standard lab method like HPLC, and compare that to a traditional drop count kit in a fresh produce matrix. And what we can see here is the high level of hydrogen peroxide in the fresh produce matrix has caused a significant false positive result on the drop count kit, but the PAA sensor has basically not been affected by that, has not been affected by the hydrogen peroxide, and is showing very strong linearity to the gold standard HPLC method. Another test we did was a third-party validation in conjunction with the customer, where we prepared a high concentration standard. And this kind of goes back to what we were saying about when you use low range drop count kits for high range concentrations. We saw that in reality during this validation. So we prepared a around 850 ppm standard, and we can see Palintest, which is the sensor technology, had good accuracy, so a nice small gap between the true concentration and our concentration, and had really good precision, which is that black error bar that you see. We had 3 leading industry drop count kits, and all of them underreported, so much worse than others, and they tended to have much greater variability between one test to the next as well. So that's the chemistry side. We can validate that we have made those chemical improvements, that we are more resistant to interferences and more resistant to underreporting. But how do we prove that we really have eliminated the technique dependency? So we worked with some customers again. On the left was one of our early trials where we went to a facility, and the blue bars on me. So I'd be considered an expert user with lots of experience in using Kemio, and the orange bar is a QA tech who had never used Kemio before in their life. And we went around their facility and we took multiple samples, and we were able to demonstrate that 2 different instruments, 2 different sensors, 2 different people showed great correlation between results, and we really did eliminate user variability. But that was one day, one point in time. So we also worked with a customer late last year for a multi-week extended trial, and we were able to show that without actually any involvement from us, the production team and the QA team were able to line up their results over many weeks of testing. So again, 2 different teams, never mind 2 different people, multiple people, 2 different teams, 2 different instruments, showing fantastic correlation in process. And one of the complaints we always get, when we talk to people, why did you choose Kemio? Why did you move to this platform? Production and QA not agreeing on what the number is one of those things that just seems universal in many food and beverage facilities and processes. So it was really great to see that, and to see how powerful it is to have those 2 departments now agreeing on what the concentration really is. So understanding what we're doing now and understanding the limitations of our current technology is really the first step to then making improvements. And I know we've only really covered the drop count kit. We've not had time to cover everything else. With colorimetry, we're measuring light, so it's very sensitive to turbidity and color. Often the range isn't very good, so we need to do big dilutions, which can cause errors. For test strips, they're very low resolution. Is it 50 or is it 90? Somewhere in between, we don't know. And they're very time sensitive. Something people don't always realize. You really have to read those strips within 10 seconds of dipping them, because that color will continue to develop over time, and you can end up thinking you have a lot more than you really do. So understanding those limitations really is the first step to moving forward, and then advancing not only our analysis, but our control of those disinfectants. And I want to go back to these graphics here. Why do we do it? And these challenges on safety, on shelf life, on reputation, and also on usage and cost, well, all of these challenges are opportunities. If we spend the time and we invest in improving our data and improving our control, all of these things will improve along with it. So with that, I'd like to wrap up. Thank you very much for listening. I really appreciate it.